Okay, good morning, everyone. Welcome to our latest webinar. The title of this webinar is Measurements of Carbon Assimilation, Reveal Competition Asymmetry in Wetland Invaded by Phragmites Australis. This webinar is being put on by Dr. Rebecca Rooney, who is a wetland ecologist and associate professor in the Department of Biology at the University of Waterloo. Rebecca's research program focuses on the response of wetland communities to natural and anthropogenic disturbances. She has published over 50 peer-reviewed papers and given hundreds of presentations to scientific and lay audiences. Her comprehensive research into the effects of common reed invasion in coastal wetlands on the Great Lakes has enhanced our understanding of the effects of biological invasion on plants, invertebrate communities, carbon and nutrient cycling, bird communities, and their foraging behavior. She has also investigated the recovery of species at-risk plants and birds following common need management. Her current studies focus on the recovery of native plants and ecosystem functions following herbicide application and insect-based biocontrol to eliminate common reed, including developing invasion-resistant native seed prescriptions for active restoration. So I'd like to introduce Dr. Rebecca Rooney and take it away.
Thanks, Tim. So today I'll be sharing some research results from a study that I carried out with my PhD student, Courtney Robichaud, to try to better understand why European common reed, known in Latin as Phragmites australis, subspecies australis, is so successful at replacing our native wetland plants. So European common reed came over, we think, from the United Kingdom over 100 years ago. And judging by herbarium samples, it arrived in Canada and Nova Scotia by about 1910, and it spread rapidly from there. It only took 10 years for it to spread across Nova Scotia and down the St. Lawrence River to Quebec City and Montreal. And it reached southwestern Ontario around 1948. It remained restricted in this distribution throughout the 1970s, but then leapt northward and westward, expanding rapidly during the 1990s, especially in the Great Lakes Basin, where it remains extremely abundant. As you can see, it now reaches from the Atlantic to the Pacific, from the Gulf of Mexico to northern Alberta. And on its march across the country, it has received the reputation as Canada's worst invasive plant, and for good reason. Scientists don't generally like to anthropomorphize, but after nearly a decade of studying common reed, I can tell you it's a real jerk. What traits make it such a successful invader? It's extremely productive. It can grow up to 5 or 6 meters tall in dense monocultures of stiff canes. Walking through it is something like trying to walk through wicker furniture. As you can see here, it grows so densely, up to over 100 stems per meter squared, that it really intercepts a lot of light. So it's very dark in there. There's not a lot of space or light left for other plants. It also creates this thick litter layer, which can impede other plants from sprouting, and birds, turtles, and amphibians can struggle to move through it. Like many invasive plants, it produces hundreds of wind-dispersed seeds that let it spread far and wide, but it can also expand rapidly from stolons and rhizomes, as well as plant fragments. Through that network of rhizomes and stolons, the clonal ramites, the individual stems here, can subsidize one another, sharing carbohydrates and responding rapidly to any disturbance. And because it's a perennial, its shoots can get started early in the spring, helping it outgrow annuals that are trying to start from seed. It is even thought to be allelopathic, releasing gallic acid into the soil, which can suppress the growth of competing plants. Another common trait of successful invaders that common reed shares is that it has a very wide environmental tolerance range. It can grow in moist soil up to water over 1 meter deep, from saltwater to freshwater, coarse textured sand, clay, or organic peaty soils. So this wide habitat range lets common reed come into competition with a wide range of native species. The only real restriction on common reed is that it's very nitrogen hungry. It doesn't grow well if nitrogen is in short supply. It has a very inefficient use of nitrogen. Nitrogen is an essential element needed by all plants and animals. A major component of its use is bound up in amino acids, as it is a building block of proteins. And so nitrogen is important for all tissues, but especially concentrated in protein-rich tissues like seeds. It's also important in the regulating the growth and development of plants, as well as in nutrient and water uptake in the root system. But a key use of nitrogen in plants is as part of the photosynthetic apparatus, being a key component in chlorophyll. Now, many plants will use nitrogen very efficiently and put more of it in the sun-receiving leaves than in shaded leaves. And this is true of at least one of common reed's main competitors, cattail. But researchers Hitterter and Potts found that common reed is not very strategic in its allocation of nitrogen. Unfortunately, human activities like farming, driving cars, etc., tend to result in a lot of available nitrogen. And so wherever humans go, common reed is usually able to follow. An influential paper by Andrew McDougall in 2009 applied coexistence theory to help us understand why some invasive species have such a high impact on the communities they invade. He described how there are these two components, niche differences and fitness differences. So the niche is really about the range of conditions under which a species can live. And if the niches of two species overlap a lot, then we expect they will compete intensely for the same handful of resources when they grow together. But that doesn't mean that one species will win out over the other. And this is where the fitness differences come in. So if we're thinking about fitness in an evolutionary sense here, if the two species have really different niches, then they may be able to coexist even if one is a superior competitor over the other. But as their niche differences diminish, as they begin to overlap, the competition intensifies, and the effect of any even small fitness difference can grow in importance. So if common reed is a superior competitor, it will begin to win out and exclude the resident species that it competes with, which it has niche overlap with, and exclude those resident species. The converse is also true. If other species have a fitness advantage over common reed, then they could keep it from invading. A study by Wilcox et al. Found that common reed was primarily replacing meadow and cattail marsh in the Long Point region of Ontario, where we do a lot of our wetland research. This area is a biodiversity hotspot, a Ramsar wetland, a World Biosphere Reserve, and an internationally recognized important bird area. It has pretty much every accolade that a wetland can receive. Both of these habitats, the meadow marsh and the cattail marsh, are highly productive and have these indicator species that are actually pretty similar to common reed. They're C3 photosynthesizing perennials with rhizomes. The meadow marsh is dominated by Canada blue joint grass, or Calamagrostis canadensis, as well as in wetter areas by water sedge, or Carex aquatilis. And these usually grow up to about 1 meter tall. In cattail marsh, we find cattails as the dominant species, and this can range from about 1.5 to 3 meters, nearly as tall as common reed itself. And so keeping in mind this difference in niche overlap, as well as fitness differences through our coexistence theory, we asked 3 questions. First, we wanted to know, does common reed have higher photosynthetic performance than these resident species that it's encountering when it invades? Does competition influence the carbon assimilation in these species equally? Both of these questions reflect the idea of these fitness differences. And then our third question, reflecting niche differences, we wanted to know how much niche overlap there is between common reed and each of these 3 resident species. To address these questions, we established a clip plot experiment in Long Point. We identified rametes that were coming up early in the spring. So remember those individual stems coming from the clonal genetes. And we wanted to use them as what we call phytometers, or test plants. So we set up these pairs of phytometers that were paired by height, and then we randomly assigned one member of each pair to a treatment with competition or without competition. And so in this case, the competition was above ground, interspecific competition. Here, for example, we have a Canada blue joint grass phytometer growing with no above ground competition, or growing in competition with Phragmites, or a common reed. And because all 4 of our phytometer species are capable of clonal subsidy through the rhizome network, at the start of the season, we also severed any below ground connections around the perimeter of the plots using a handsaw to a depth of 50 centimeters. So as I said, competition was interspecific. So for example, here we have water sedge. If it was the phytometer, it grew either without above ground competition or in competition with common reed, and the same is true for the cattail and for the Canada blue joint grass. Common reed we grew in combination with each of those 3 resident species. So we had 10 different treatments total, and each had at least 12 replicates established each year. We repeated this experiment for 2 consecutive years. And we had to double the number of replicates in water sedge and Canada blue joint grass because those plants are more delicate, and we needed to make sure that enough survived the growing season to let us take our measurements. We maintained these treatments, visiting the sites every 10 days to measure the height of the phytometers and to reclip the growth in the no competition treatments. To characterize fitness, we measured carbon assimilation and created photosynthesis radiance curves using our Cirrus-3 and our PLC3 LED light unit with a narrow leaf cuvette. We selected fresh entire leaves in full sun exposure and repeated the measurements during peak above ground biomass in July and August. To relate photosynthetic performance to the foliar nutrient contents, we also collected plant tissues from each phytometer, which we analyzed for carbon and nitrogen content, as well as for the isotopic composition of delta-13 carbon. So delta-13 carbon tends to be higher in plants which are photosynthesizing more efficiently because they exhibit less discrimination against the heavier carbon-13 isotope during stomatal diffusion and carboxylation by rubisco. So in terms of the niche differences, we tried to quantify these by selecting 15 sites that were dominated for each of the 4 phytometer species types. And at each of those 15 sites, we collected a 10 centimeter deep core to measure a variety of important plant nutrients, as well as soil moisture. And we measured the incident light coming in that was intercepted by the canopy. So here I am on top of a 6-foot tall ladder, and I've got my light meter stretched out above the canopy to measure incident radiation or photosynthetically active radiation. And then simultaneously down here at the base of the canopy, you can't even see it, in the shade there, a measurement is being taken. So we can measure the proportion of incoming photosynthetically active radiation that's intercepted. And we found that the interspecific competition affected the resident species more. Carbon assimilation was actually greater in common reed than resident species at full sun. So these are the resident species. Full sun was at 1,500 micromoles per centimeter per meter squared. And we have our 3 different resident species compared to the common reed grown either without competition or in competition in our pairwise crosses. So overall, common reed had more carbon assimilation, but also the effect of that interspecific competition was greater in the resident species than in common reeds. We saw more of a reduction in carbon assimilation at maximum irradiance. And we published these results in a paper, which I can share with you later if you want to contact me. The photosynthetic efficiency of common reed, as indicated by the delta-13C values, was also greater than that of the resident species. And it was less affected by above ground competition than in the resident species, which showed more of a drop in the delta-13C in those phytometers with competition. So the resident species were less efficient in their photosynthesis when growing in competition with common reed, and common reed did not see its photosynthetic efficiency affected by that competition experiment. Total carbon, this middle panel here, was not really affected by the competition treatment, but nitrogen differed quite a lot among the species. It was highest in common reed on average and lowest in the water sedge down here, and in Canada blue joint grass. And they also had lower nitrogen content in their leaves when grown in the competition treatment compared to when grown without competition. Cattail didn't really seem to show the same pattern. Common reed also had the biggest niche region, reflecting its broad environmental tolerance range, where Canada blue joint grass had the smallest niche region. So we summarized all the niche information with a principal components organization, which revealed that the resident species are actually exhibiting resource partitioning. So we see Canada blue joint grass kind of here on the left with very little overlap with water sedge or with cattail. They're kind of spread apart along this first ordination axis. And then common reed is overlapping with all of them with a much larger niche region in total. So this strategy of resource partitioning helps the resident species achieve coexistence by minimizing the degree of niche overlap. But common reed exhibits this high overlap with all 3 of the resident species, especially the water sedge. You can see it's almost entirely enclosed in the larger sort of polygon here representing common reed. So there's not really any refugia where it can escape competition. We use this neat R package called niche rover to calculate the niche region size and the degree of overlap in niches among the phytometers. And interestingly, the degree of overlap varied. So cattail actually only had about 50% overlap with common reed, whereas water sedge had the most overlap, over 90%, and Canada blue joint grass only about 83%. But because the niche region of common reed is so large, these overlaps are asymmetrical. So the intensity of competition would be greater for these resident species than for common reed, as common reed has more of this refuge in its niche where it can avoid competition through resource partitioning. So European common reed is invading cattail and meadow marsh, where it displaces cattail, water sedge, and Canada blue joint grass, among other important wetland plants. We were able to shed light on why this is by quantifying both the niche overlap and fitness differences among these phytometers. And this clearly fits into our framework for coexistence, landing squarely in this box here, where common reed is excluding resident species. It had higher photosynthetic efficiency and higher carbon assimilation rates and was less affected by competition than the resident species. Now, if there had been greater niche differences, we might have seen more possibility for coexistence. For example, cattail, where the niche overlap was only about 15%, for cattail, we'd expect more coexistence to occur than with Canada blue joint grass and water sedge, where the overlap was over 80%, so we're more over here. It's therefore not surprising that those two species evidence the greatest reduction in carbon assimilation rate in response to competition treatments with common reed, whereas common reed's carbon assimilation rate was hardly reduced at all, even when it was in competition with cattail. Consequently, we know we need to prioritize keeping common reed from invading these vulnerable meadow marsh and sedge meadow habitats where it will face little resistance from native plants, whereas cattail we can expect might put up more of a fight. We can also use this information to identify traits that we want to incorporate in seed prescriptions for wetland restoration to encourage the development of a plant community that will be more resistant to invasion by common reed. For example, species with fast growth early in the season, or species that are able to rapidly take up nitrogen. Indeed, this is our next step in the research we're conducting. We're using plant functional traits to identify plant community members that might be better able to coexist with common reed and enhance the natural biological resistance of wetlands to invasion. Well, none of this work could have been possible without a large group of project partners, including NGOs, government agencies, and private companies. Through their support, I've been able to train 18 students in wetland ecology through this and other related research in Long Point. In particular, I want to thank those students who assisted Courtney and I in this very labor-intensive study. And I will draw your attention to my contact information here at the bottom if you would like a copy of the paper that I mentioned, or if you have any other questions we don't have time to discuss today.
All right, terrific, Dr. Rooney. That was a fantastic presentation, and thank you very much. We'll now open this up to questions from any of our attendees. If you have any questions, feel free to ask or put them in the chat, and Dr. Rooney would be happy to answer. All right. Okay, well, if there's no questions, well, I mean, if anyone has any questions, you can feel free to email or contact us here at PP Systems or Dr. Rooney directly. We'll be sure to have this webinar posted to our website soon for later viewing if you wish. Otherwise, thanks everyone for attending, and have a wonderful afternoon.
Thanks, Tim, and thanks, PP Systems, very much.
Thank you, Dr. Rooney, as well.
Thank you for attending this webinar. To learn more about how the CIRUS-3 portable photosynthesis system can enhance your research experience, visit ppsystems.com.